So hello and welcome back to Excision Academy. So we were doing DNA repair, right? So in the previous lecture, we discussed about the nucleotide excision repair. And today we are going to discuss about the recombination repairs. So this recombination repair is required whenever there is a DNA double stranded break is there. In such cases, we can have the recombination repairs. Okay? So they are going to repair by retrieving the sequence information from the undamaged DNA. तो ये किन के बीच में हो सकता है होमोलोगस क्रोमोसोम्स के बीच में हो सकता है राइट सो लेट्स सी हाउ इट इज बीइंग डन so excision repair uses undamaged DNA strand as a template to replace damaged segment of DNA on the other strand correct so if you remember कैसे था nucleotide excision repair में like you had the strand over here right यहाँ पे कुछ problem थी so they have nicked over here and removed these particular nucleotides then this was removed this entire strand was removed then the polymerase will come and ligase will come reseal the nick ठीक है Akshaya so we have just started this is the first slide only so we are talking about like in this session today we are going to discuss the double strand repair how it is being done that is the recombination method okay so the recombination based pathway retrieves sequence information from the sister chromosome so in this how like पहले हमने क्या पढ़ा था कि वो आपका टेम्पलेट स्ट्रैंड यूज करके रिपेयर करता है in this case in the recombination repair method it is using the sequence information from its sister chromosome okay okay so the DNA recombination it also helps to repair errors in DNA replication it is helping in the DNA recombination helping in repair in DNA replication whereas recombination helps और कहाँ पे इसका काम आता है तन्वी yes where do you see DNA recombination happening
meiosis
हाँ so जहाँ पे you are going to have the homologous chromosomes having the synapse formation and recombination is there right so that's how you get the genetic diversity correct now along with this we have got the recombination happening at the repair level also okay now consider a replication fork which encounters a lesion in the DNA let us consider again the example of thymine dimer and now it has not been corrected by the nucleotide excision repair so मैंने आपको पहले भी बोला so we have got several types of repair mechanism एक नहीं हुआ so then on the downstream we have got the another इससे भी नहीं हुआ तो तीसरा चौथा पांचवा now if still it is not able to repair the DNA then the problem is that either the cell will undergo apoptosis or it will take the fate of being cancer okay so we do have checkpoints we do have got this repair mechanism it just depends on these methods how efficiently they can repair the DNA okay so now let's consider that there is a DNA which is having a lesion let us consider the example of thymine dimer okay which has not been corrected by the previous mechanism of nucleotide excision repair now what will happen the DNA polymerase will sometimes stall attempting to replicate over the lesion. So what does it mean? It means DNA polymerase now रुक जाएगा, ठीक है? It will get stuck over there. Now, although the template strand cannot be used, the sequence information can be retrieved from the other daughter molecule of the replication fork by recombination. Okay. Now, once this recombination repair is complete, the nucleotide excision system has another opportunity to repair the thymine dimer. तो बस अभी क्या समझ में आया हमें that अगर हमने इसको भी एक और मौका दिया we have given another chance to nucleotide excision repair method to go for the repairing once again कैसे so if suppose there was a mistake okay template में एक thymine dimer था DNA polymerase is not able to continue the replication it will get stuck over there now the sequence information will be taken from its daughter molecule okay so let's see how it is being done So mutants which are defective recombinations are also known to be sensitive to UV rays. अब क्यों UV rays हम बोल रहे हैं यहाँ पे? We have taken the example of thymine dimer, right? So वहाँ पे आपका nucleotide excision repair काम नहीं कर रहा है. Now your recombination method is also not helping in the repairing. So these mutants, okay, these strains become the sensitive to UV light. So they become the sensitive mutants. Now consider a situation in which replication fork encounters a nick in the DNA template. This is your second scenario. Okay, so what will happen? So the passage of the fork over the nick will create a DNA break, repair of which can only be accomplished by double strand break repair pathways. ये समझ में आया? हाँ, कि अभी आपके पास डीएनए है, ठीक है, एंड यहाँ से आपका रेप्लिकेशन फॉर्क आ रहा है. सो यू हैव गॉट ऑल योर डीएनए पॉलीमरेज, यू हैव गॉट योर डीएनए पॉलीमरेज कमिंग, यहाँ से हेलिकेज उसको अनवाइंड करता हुआ आ रहा है. सो व्हाट यू सी, यहाँ पे आके एक निक है, ठीक है. सो निक मींस हियर यू हैव गॉट द, ओहो सॉरी, सो हियर यू हैव गॉट द ब्रेक, ऐसा. You can see there is a gap, right? Now this can be only repaired by this double strand break. अब हम कैसे करेंगे वो हम देखते हैं. Okay, so double strand breaks in DNA are also repaired by direct joining of broken ends. So simple mechanism क्या होगा? That your DNA ligase comes and reseal the gap, right? Reseal the nick which is being present over there. Are you guys able to follow? Akshaya, Tanvi?
Yes, ma'am.
Okay, okay. Huh? So the
Yes,
double strand
ma'am.
break is there, yes, which is most cytotoxic of all kinds of DNA damage. क्यों? Because अगर आप किसी भी double stranded, sorry, जो DNA break है उसको अगर आप ऐसे ही छोड़ दोगे, so what will happen? The chances are that your when forming a chromosome, there will be a loss of the part of the chromosome. Okay. Now, when there is a loss of the chromosome part, what will happen? Either it will go for the apoptosis or it will have the cancer formation. Okay. Neoplastic transformation is also otherwise known as cancer. So cells typically have multiple overlapping pathways of coping up with the DNA damage. Okay. So the effective method is that the sister chromosome will provide the template for the precise restoration of the original sequence across the site of the break. For example, in the case of yeast cells, recombination-based DSB repair is a principal pathway. Okay. Now, but what happens early in the cell cycle before two sister chromosomes have been generated by DNA replication? Okay, अभी इन्होंने क्या सिनेरियो दिया है कि यहाँ पे अभी सिस्टर क्रोमैटिड्स कैसे बनेंगे, अभी रेप्लिकेशन हुआ नहीं है. Okay, so you know that once the DNA replication gets completed, then only the sister chromosomes will form, and during the mitosis they will get separated. Correct? But what if it is happening before the DNA replication, or sorry, before the two sister chromosomes had been generated? अगर क्या करोगे अगर आपका ये पहले break हो गया तो? Okay. Now, if
Ma'am, your
हाँ,
voice is breaking a lot.
yeah, okay, but it shouldn't actually today. I'm using Wi-Fi now also. Hello, it is breaking now also.
Yes, ma'am.
One second. Huh? Okay. So the question was like, if the nick of this problem, okay, this DNA break is happening in the cell cycle before the two sister chromosomes have been generated by DNA replication, तो क्या करेंगे? ठीक है? So if still unreplicated chromosome suffers a break, then no sister chromosome is present to serve as a template in the recombination based repair pathway. तो क्या हो गया? What does it mean? These two sentences? अगर टू क्रोमोसोम्स यानी कि डीएनए रेप्लिकेशन से पहले अगर ये प्रॉब्लम आ रही है एंड विच इज नॉट गेटिंग रिपेयर, देन व्हाट विल हैपन? So now you don't have a template, now you don't have a sister chromosome so that उसके sequence को देख के हम repair कर सकें, right? Now we can't do recombination based method repair, so that cell is not going further then. Okay, so this is the answer for that. Okay. But now that is one scenario where it is not getting repaired. So now what we are going to do is we are going to take the alternative method, like by which though the sequence is not going to be the same, okay, but it is going to make sure that the cell is alive. Again, the same analogy, like मरने से हाथ पैर टूट जाए वो better है ना? So same way, even though the DNA is not having the same sequence it is carrying, but it is still alive. So thinking that later it can be repaired. Okay. So the another method which comes into the scenario is non-homologous end joining. इस पे based questions आए हैं. Okay. So if you ask me like in the DNA repair system, basically what are the important topics, then that is your about the thymine dimer, then mismatch or nucleotide excision repair, and then after that NHEJ. So in सबको अगर आप अच्छे से पढ़ लोगे, so you can claim and you can say that yes you have done with your DNA repair mechanism. Okay. Okay. So in NHEJ, it's a backer system in the case of yeast, and in higher cells, it is a principal pathway. Okay. So the machinery for performing NHEJ protects and processes a broken end and joins them together. Okay. So because this sequence information is lost from the broken ends, the original sequence across a break is not faithfully restored. So what does it mean? अगर आपका break हुआ है, okay. So non-homologous end joining में क्या होगा? Even though it is not between the homologous, right? What will happen? The DNA will be joined. Okay? इतना idea आ रहा है कि आपका अभी sequence information is lost, but the chromosome is not damaged after the rejoining. Okay? So this is mutagenic. Of course, the mutagenic consequences of NHEJ mediated DNA end joining are far less hazardous to the cell, that is having the cell with the broken DNA. Okay, which is not repaired yet. Same analogy that let's have the wrong sequence of the DNA, wrong sequence of the DNA as compared to having a broken ends of the DNA. Okay, so this is the concept that we have kept. Now let's see how it is being done. So what is the mechanism behind all these things? We will see in the figure. Okay, so this is how it is being done. So can you see the figure properly? Can you see the names of the proteins here? So they are like Q7080. Okay, can you see that they are going to recognize the broken ends of the DNA? So it's a heterodimer of 7080. So what is the function of Q7080? That they can identify the broken ends of the DNA. Okay. Later, once they identify the broken ends of the DNA, these are the proteins. Now what they do is they are going to recruit the protein, or the kinase is called DNA PKCs. In turn, these DNA PKCs is going to recruit protein called Artemis, which is Artemis is what? It's an exonuclease and endonuclease activities processing enzymes, which is going to process the broken ends of the DNA. Now, a complex of ligase 4 with along the XRCC4 and the another proteins, they are going to join the broken ends of the DNA. Is this pathway is clear? Yes, Tanvi?
Yes, ma'am.
Okay. I hope the voice is coming now properly.
Yes, ma'am.
Okay, okay. So I'll just repeat once again. Okay, so here, this is your NHEJ, non-homologous end joining of your DNA. So यहाँ पे DNA में क्या हुआ है? आपका double strand break हो गया है. Okay. Now we just need to repair this. अब ये कैसे होगा? So you have got the proteins called Q7080, which is going to recognize the broken ends of the DNA. This is going to recruit your DNA PKCs, which is later going to recruit your Artemis. Artemis क्या है? आपका exonuclease और endonuclease activity वाला enzyme है. Now what they're going to do is they just chop off the randomly Q nucleotides from here, okay, from here and here. Later the ligase will come and reseal the DNA. तो अभी इस process में क्या हुआ आपका? Few nucleotides are lost, okay. So what does it mean? अगर यहाँ से कुछ nucleotides गायब हो गए हैं, is it going to affect the DNA sequence or not? What do you think, Tanvi? Is it going to affect the DNA sequence or not?
Yes, ma'am.
Yes, because few nucleotides you have removed, right? So क्या पता वो gene कम हो गया हो, ज़्यादा हो गया हो, so it is going to affect. Okay, but if that gene is not, what do you say, it's so important for the organism, then it is not going to be lethal. Just imagine that it was very important for the organism and its expression is required for the survival of the organism, then it is going to be lethal. So this is not repair. It is again it is going to cause the death in the organism because there won't be any gene expression of that particular strand, and then later the proteins or the mRNAs which are required will not be expressed, and hence the organism might die. Okay. Okay. For example, it is not that frequent in the case of bacteria, but it is present in the mammalian cell a lot. Now, an example is there of the Bacillus subtilis. Okay, so the Bacillus subtilis produces a Q-like protein and a DNA ligase when it sporulates and packages the proteins into the mature spore. So we know that these are the certain bacteria which can form the endospore. आपने microbiology में पढ़ा होगा that there are certain bacteria which can form the endospore. Do you know what are endospores? Yes?
Yes, ma'am. Which
Okay.
are
Okay.
produced outside.
हाँ, so it's like a protective mechanism of the bacteria in the harsh conditions. Okay. तो आपको ये बैक्टीरिया होगी, उसके ऊपर एक sporulating structure आ जाता है which is protecting the bacteria from the harsh conditions. And whenever a good and favorable environment is present, then they start germinating again. It means they are just coming out of the spore then. So this Bacillus subtilis, okay, it produces Q-like protein in DNA ligase when it sporulates and packages the proteins into the mature spore. So this Q and the DNA ligase, which is representing a simple 2-protein NHEJ system, repair the DNA breaks when the spore germinates. Now, if suppose there are the mutant spores which lack these proteins, a spore of the Bacillus subtilis not having these proteins, they're highly susceptible to dry heat, a condition which is known to break the DNA. So we know that. See, that's why in the microbiology, if you know, we do autoclaving. So you know the principle of autoclaving, right? So we are going to, we give the moist heat of around 121 degrees Celsius for around 20 minutes, right? किसी cases में हम 30 minutes भी रखते हैं with the 15 lb pressure. So these are the requirements of autoclaving. To say that yes, you have done the proper autoclaving. This autoclaving means what? You are going to sterilize your media or whatever the utensils required for your experiments. Correct. Now, there are certain bacteria which results in the formation of endospores. So these endospores are not they are resistant to the heat conditions. So ऐसे cases में क्या होगा? If the bacteria is not having these proteins like Q proteins and the DNA ligase 4, if they are not having, then they become susceptible to the heat. Okay. So this heat will then result in the DNA break. Ab DNA break ho gaya, so obviously the organism is gonna die. Okay. So upon germination, if heated mutant spores are unable to resume the growth because they are unable to rejoin the heat-induced break, and hence the bacteria will die. Okay. Is this concept is clear? Like how the NHEJ in bacteria is helping in order to become the resistant towards the heat. Yes, Anvi, is it clear? Is it clear to you?
Ma'am, can you repeat once again?
Okay, I'll just repeat. Okay, so we are talking about NHEJ. Okay, so it is ubiquitous in eukaryotic organism. It means it is normally present in eukaryotic organism, but in the case of bacteria, in few cases only, this kind of repair mechanism is present. Now, let us take the example of Bacillus subtilis, which has a potential to form endospore. Okay, so endospores are formed in order to become resistant to harsh conditions. Now, what can be the harsh conditions usually a bacteria is facing? The heat, okay, or you can have the chemicals or the pressure, high pressure. So these kinds of harsh conditions, whenever it is present, there are few strains of bacteria like Bacillus subtilis, they can form the endospore. Okay, now what is the mechanism? Like how the endosperms are resistant to heat? It is because heat, we know that heat can cause DNA damage, okay, DNA breaks. Now, if the organism is having your Q protein and the DNA ligase 4 protein, then they are able to repair whenever this heat is causing the DNA break, and hence they become resistant to heat. Okay. Now, imagine a situation where you don't have a normal Bacillus subtilis endospore forming. You have a mutant. Mutant means this bacteria is not having this Q protein and the DNA ligase 4. Okay, they are not having. So what does that mean? If I give heat to such bacteria, such mutant bacteria, which is lacking these two enzymes or these two proteins, what will happen? They will become susceptible to heat and hence cannot grow further. Okay, now it is clear. So whenever there is a germination happening and if suppose there is a heat present or the high temperature is present, these bacteria cannot grow and they will die. Is this much clear?
Yes, ma'am.
Okay. Okay. So this germinating spores. Now this was a question which came in the exam, like how the bacterial spores are repairing their DNA whenever there is a heat, and also you can just say that now it is the answer is neighbor, I'm saying non-homologous end joining. Okay. Now they have only one chromosome, therefore they cannot rely on their sister chromosome. So this can be asked in the interview also कि आप generating spores को non-homologous end joining से repair कर सकते हो मगर double strand break से क्यों नहीं repair कर सकते? Then you can say that in the case of bacteria you don't have sister copies, right? You just have only one chromosome. So we don't have a sister chromosome onto which the sequence information is present and you can rely on that. Okay, so you can say such answers whenever such questions are being asked. So the spore chromosome is tightly coiled into a remarkable donut-like structure and could not hold the ends of the breaks in the DNA. And this close juxtaposition could facilitate correct rejoining of ends even if the chromosome has multiple breaks. So you can see if the chromosome is tightly packed like this, okay, kitne bhi break aane do because of heat. Since they are in close proximity to each other, it becomes easy for proteins like Q and ligase 4 to repair them. Okay, so it's not that आप ऐसे मत सोचो कि जब हम हीट देते हैं तो वो हीट सिर्फ एक ही जगह पे डीएनए डैमेज करेगा. It can be multiple places. So you have multiple proteins which is acting on the repair mechanism. Okay, so just imagine a situation where you can see so many chromosome breaks are happening because of the heat, high temperature, and this is also activating your NHEJ repair method. So those many proteins will get activated and they are going to repair the DNA strand damage. Clear? Okay. Is this much clear?